I'm here with Stefanie Dimmeler, who is Director of the Institute of Cardiovascular Regeneration at the University of Frankfurt, and she's also Chief Editor of EMBO Molecular Medicine. And welcome, Stefanie. And, for my first question, I wanted to ask you, you've had a very interesting career, and I saw at one point that you're involved in experimental surgery, so I thought it would be interesting if you could tell us maybe a little bit about your career background.
It's actually, the first decision, of course, is always what you are going to study, and I was quite sure that I wanted to study something with natural science, but that it happens that I studied biology in the end was just by chance, I have to say. And then, of course, the next major decision was what to do after the PhD thesis, or what to do as a topic in the PhD thesis. Yeah. And there I chose a more medical relevant topic, which was working with platelets, which are small blood-derived cells, which make blood clotting. And this, of course, dedicated a little bit my research towards more a medical-oriented research, although it was really biochemical pharmacology there. But this was maybe the reason why I was getting interest in medical science. And this was also the decision why I then, as a postdoc, did, against this recommendation of all my supervisors, didn't enter Max Planck Institute or something like this, but I went to a surgical department. Okay. I didn't realize how much difficult it is to really make science in such a clinical background, but it was very interesting, and I learned a lot.
You made the transition from research to translational medicine, yes, and via surgery.
Yes.
That's fascinating. So Stefanie, I'm also interested in the question about what advice would you actually give to young scientists who are just starting out and maybe who have an interest in translational medicine?
So I think that also for PhDs, it's a very attractive, let's say, career chance now in medical scientists. And if you choose the right conditions, meaning that you choose the right environment, it can be very successful. You have to be, however, aware that you don't end up in a, let's say, a place where they have good ideas but no infrastructure. Mm-hmm. And of course you can have really clinically very important ideas, but if the boss of the department is not aware of what it means at the basic science levels, that you have the novel technology standards and these things, you have to really check carefully that the environment in these clinical departments is appropriate for doing high score science. And this is the advice I would give for PhD students. Otherwise, I think it's a great chance to do clinical translatable work, and you can really learn a lot. And this combination of medical doctors and basic scientists, at least in my view, is very interesting because it opens for both really new avenues and new thinking. Mm-hmm. So maybe you learn more than if you go simply to a more very specialized lab.
And so looking back on your career, what have been some of the bigger challenges for you in making all those changes and getting to where you are today?
I think the challenges are on a daily basis. So of course, you have major step, as I mentioned before. The decision, what are you doing as a PhD thesis, clearly is one step which affects maybe people you like in the lab, which you have visited. And the second step, what you're going to do as a postdoc, of course, influences your life. Mm-hmm. And the decision to go to surgery clearly drove me in a direction of translatable science. And this was also the reason why I was then recruited in cardiology in Frankfurt, because they thought this is an ideal person which knows from a translational point of view about more the really clinical studies, but also has background in blood clotting and endothelial cells. So this brought me actually to the position where I started in Frankfurt.
So now you're working in cardiovascular regeneration and you're studying microRNAs, and I thought it would be interesting to hear about your work on biomarkers and also the therapeutic aspects of microRNAs.
So microRNAs are small coding RNAs, so they are really tiny fragments of RNA. And I think they are interesting because of two things. You mentioned already they can be used as biomarkers, so you can detect them surprisingly in the circulating blood. So you can detect, for example, microRNAs which are exclusively expressed in the heart in the circulation, which tells you if they are present in the circulation that maybe the heart is activated or injured. And although we don't know yet whether they are really better than the yet established markers, they may provide additional information about the function of the heart and maybe also the activation of heart cells. Second, these microRNAs can be inhibited quite efficiently. Right. So you can inject inhibitors in mice, for example, and they are having long-lasting effects up to several weeks. So this might be also useful as therapeutics. And there's one clinical trial already in phase 1 with targeting liver disease, where antimir's inhibitors of microRNAs are used. And so we hope that we may be ending up also in translating something in the cardiovascular field, and for example using MyCoRN inhibitors for the treatment of myocardial infarction patients or other maybe more acute diseases.
That's one of the big challenges, is actually going from basic research to the clinic. And I wonder if it's possibly a difficult question, but how do you balance the expectations of patients and stakeholders with the way science proceeds in terms of time and actually bringing benefits to patients in the clinic? So that's my question.
I think it's a very important question, a very difficult question. Particularly in the stem cell field, we have seen such a huge hope based on some really preliminary studies that patients are expecting to really, you get one cell treatment and then you stand up and you are feeling like 18 years old if you're 70. So certainly these expectations are too high, and in clinical science usually you have small developments and it takes long. So you have to first make sure that the things are safe, you have to follow up patients, and then in cardiology, for example, you need really large patient numbers to make a reliable assumption. So you have to confirm your findings from the lab in small clinical trials and then move on to larger clinical trials. So it takes a long time. And I think this time, and also money-wise, it's so expensive. A huge clinical trial takes several 10,000 millions.
It's a big undertaking.
It's not something you can do by yourself. You really need teams of people, a lot of money also to make a therapy out of a basic science finding. Mm-hmm. And sometimes, of course, as a basic scientist, you have to explain what you're doing to raise also the interest in it, and also to let people understand what you are doing. On the other hand, of course, it's very hard for patients to understand at which levels you are at the time being and what are the time frames until they may profit from the research.
Okay. Yeah, I think there's a communication side to this, as well as just educating people how long it takes to go from basic research to clinic, and that's very important.
And also how many things fail.
Yes, indeed.
Of course, we have good ideas, but all our models are also limited, and we do a lot of studies in animals, small animals, or also in experimental models, and it's very difficult to then be sure that this also have patients.
To go from animal system to testing in patients, that's an extreme challenge. Yeah. So now I wanted to switch tack a little bit, and I think since the beginning of the year you're now the chief editor of EMBO Molecular Medicine, and I just wanted to ask the question, why did you actually take on this job?
So actually I was not really thinking about taking that job because I have so many things to do already.
Sure.
I think it's very nice to have a journal which bridges excellent science and more translatable aspects, and I think EMBO has a really, really strong profound expertise and also standing in the basic science community, and then to bring this expertise into the medical translatable community, it would be really exciting and profiting also, because if you bring people together, you may also end up in getting advanced science towards translation. And I see this meeting EMBO members which do basic science questions. I think the gap is really huge, and it's important to bridge the gap. So I think this is an excellent task of EMBO Molecular Medicine to try to bring these two communities together.
Right, okay. And now it's almost been a year, so I'm very interested to know what your findings have been in the first 12 months, and perhaps what are the main highlights of the first year.
So I think the main highlight is, of course, our impact factor, which is surprisingly good. This is not my work, it's the work which had been done in the first 2 years of EMBO Molecular Medicine, but it's really, with 8.5, it's very high. What
do you see as being the biggest needs of the translational medical community in the years ahead that can actually be met by the journal itself?
The major need actually is really the transit from the lab to the small clinical trials, which is very difficult, of course, but needs to be supported by academia. I think big pharma companies are no longer investing so much in cardiovascular, and we need to bring in more molecules, more ideas that they may suggest to take this over in larger steps clinical trial, but these ideas to translate them is very important. I don't know whether EMBO Molecular Medicine can fulfill this, but certainly we can provide a forum for translatable science, so to make the community aware of very interesting potentially relevant findings which might end up in new treatments.
And then stepping back a little bit from your own research and also from the journal itself, are there any other research areas at the moment that excite you in the cardiovascular area?
Oh, stepping back, there are so many exciting things. And then when I listened today for the EMBO EMBO and the new members of EMBO presenting themselves. These are so many fascinating ideas that I really cannot talk about all of it. Of course, the microRNA community is very exciting for me. In general, also RNA biology is very exciting, but also the new imaging tools to study molecular mechanisms in the cells, like cell behavior, movement, which gives us really a three-dimensional impression of what's going on in the cells. Yeah. I think this will change our understanding of biology. And this, of course, may also in the end helps in getting better druggable targets, maybe finding things which can change these complexes. I think this is something which I am really excited about. So imaging is certainly a technique only, but this provides us, I think, with a lot of new knowledge.
Stefanie Dimmeler, thank you very much.
You're welcome.